why the heck are you? Okay, and then you'll leave me. Okay, good. All right, everybody. Please welcome up the next guest. Gabe Wunch. Well, hello, everybody. My name is Gabriel Wunch, and today I'll be discussing with you all an experiment that we conducted on the effect of lentiginous adote secretions on an acute lymphoblastic cell line. Now, cancer. Unfortunately, many of us, ourselves included, may have experienced the effects of cancer and how it just devastates the human body, as well as the treatments and how horrific they can be in affecting individuals. Growing up, we had a family friend who underwent a leukemia diagnosis, and subsequent treatments. I remember just being really marked as a young child and seeing the pain and suffering that she went through. So coming to ORU and hearing about the Ranahan Cancer Research Lab, I was interested in joining the research, and that's where this project came from. So in this project, the goal of it was to discuss and detect if there is any cytotoxic effect of lentiginous adote secretions on an acute lymphoblastic cell line. So using a medicinal mushroom secretion similar to what Diego did in his previous project. So throughout this experiment, throughout this discussion, we're going through first the background of what is cancer, what is this specific type of medicinal mushroom. We'll go through the methods and materials. So specifically, what did I do? We'll discuss the results. We'll go into some further discussion and then move on towards some potential future directions. So what is cancer? Well, cancer is a grouping of around 100 different diseases that have the same origination. Over time, with an accumulation of mutations, the cancer cell begins having an unregulated cell growth and cell proliferation. So they keep on growing and growing and dividing. Now, this genetic transformation often happens in these specific genes that affect cell growth. So, for example, we have our proto-oncogenes. Now, if a proto-oncogene becomes mutated, it's similar. It's analogous to the accelerator pedal on our car. Like we're going 65 miles per hour down the freeway. We have our pedal a certain distance down, but if it becomes mutated, it becomes an oncogene. And these proteins, it's very similar to as if it's pedal to the metal, foot all the way down, just continually grows and grows and grows. Then we have our tumor suppressor genes. This is analogous to our brake on our car. Now imagine you're going 65 miles per hour down the freeway. All of a sudden you don't have a brake and you can't stop. That's similar to what happens when the tumor suppressor genes become mutated. So what causes these mutations? Well, we have both intrinsic and extrinsic events that can cause these. So intrinsic events, when these mutations happen, there can be mistakes by the DNA repair mechanisms. And when these mistakes occur, the mutations stay. We also have extrinsic events, so various different carcinogens, things in our environment. So we could have environmental pollutants. We could have obesity as one, smoking, different viruses. All of these can affect and cause cancer. So the specific cancer that I chose for this experiment was an acute lymphoblastic leukemia cell line, otherwise known as ALL. It is specifically a cancer of the bone marrow and the blood. Now, normally within your bone marrow, hematopoiesis occurs. This is essentially where we have our blood stem cell, as you can see right here, that begins producing lymphoid stem cells, and then we get our lymphoblasts, which result in our B and T lymphocytes. Lymphocytes, just a fancy word for our white blood cells, a type of immune cell. However, when these cells become cancerous, we get an excessive amount of these lymphocytes within the blood. They begin crowding out healthy cells and causing problems throughout the whole body and disrupting the normal process of blood cell formation with hematopoiesis. And this can spread to the central nervous system, so your brain, your spinal cord, your spleen, your liver, your lymph nodes throughout your body, and other organs. So who's affected by ALL? Unfortunately, around 80% of ALL cases occur in children. They're pediatric cases. But even with these, it's found that in pediatric cancer cases, around 25% of these cases are due to ALL, making it the most common childhood malignancy. In adults, around only about 20 to 40% of adults who receive ALL treatment ever receive long-term remission. So in the grand scheme of things, it can be a pretty fatal disease, though there has been success with pediatric cases receiving a 90% of long-term remission. So how is cancer treated? Since the 1960s, so for 60 years, we've had the three mainstays of treatment. We've had chemotherapy, radiation therapy, and surgery. Now, chemotherapy is essentially a drug that kills fast-growing cells, both the cancer cells, but also killing your own cells. And now, then another treatment option we have is radiation therapy, in which we actually aim to induce mutations and destroy the DNA of these cancerous cells and hopefully cause cell death, and surgery removing those specific tumors. Now, how is ALL treated? Well, in ALL, it deals with a severe chemotherapy treatment that's very, very aggressive, as well as CNS preventative therapy. What is that? Well, essentially, leukemia can travel to the brain, and the brain and the spinal cord can become a sanctuary site to keep these cells continue to grow and continue to divide. So to prevent this, they actually will insert a reservoir into the brain, to the ventricles of the brain, and chemotherapy will be applied directly to the brain, as well as radiation therapy to the brain. So pretty extensive treatment. And because chemotherapy can devastate the human body and removing the hematopoietic stem cells within the bone marrow, often these patients need to receive a transplant from a donor of different stem cells to go into their bone marrow. And in these transplants, many different complications can occur. So in ALL treatment, there's many different side effects. One is treatment-related mortality. So a treatment-related mortality is essentially when the cancer doesn't kill the person, but the treatment, the way we're actually approaching the cancer, is what kills the individual. And in a study done in Ontario, Canada, between 2003 and 2012, they found that around a quarter of all pediatric cancer deaths were caused by the treatment and not by the cancer. And one of the highest culprits, leukemia. And they believe it's because of the stem cell transplants as well as the chemotherapy method. It is very, very aggressive. And even with the stem cell transplants, graft versus host disease occurs, where these new cells begin attacking the organs of the individual who receives the transplant. And some studies have shown between 30 to 70% of these individuals that receive the transplant can get graft versus host disease. We also have short-term side effects of cancer treatments in ALL. So for instance, hair loss, nausea, low amounts of red blood cells, just causing so much pain to the individual as their own healthy cells are killed as well. We also have late effects. Because of the toxicity of this treatment, individuals can have premature mortality. They can die younger than they would have normally because their organs were damaged by the treatment. So this begs the question, can we find a cancer treatment that is non-toxic to the human body while also selectively killing cancer cells? And this is where we get the medicinal mushroom. Now, for thousands of years in Near East medicine, the mushroom has been used, and recent studies have demonstrated that the immunomodulatory processes, the anti-inflammatory processes, as well as anti-cancer effects in mushrooms continue to be demonstrated in vitro and clinical studies. And it's because of this, because of demonstrated in clinical studies, specific triggering and inhibition of cancer responses, that it's become an approved adjunct to cancer therapy within Japan and in China. So specifically for my experiment, I chose the shiitake mushroom, both tasty and hopefully has anti-cancer effects as well. We have in many different extracts they found. So essentially a cancer extract is where they take this mushroom extract, they take the mushroom itself, they break open the cells and find what's within these cells. In these extracts, one of the most biologically available molecules they found was a molecule called lentinin. And lentinin in vitro studies demonstrated a direct cytotoxic effect against various different types of cancer, So mushroom secretions. Why mushroom secretions instead of extracts? Well, many of these studies that have shown anti-cancer effects with mushrooms have dealt with the extracts of the mushroom, where again, like I said before, you break open the mushroom in order to get these molecules. Well, turns out a majority of the molecules that are within the mushroom are actually made for the mushroom and not made for us. So what about mushroom secretions? Well, mushrooms, because they're not like us. We take in our food's broken down and then it's digested. Mushrooms, they have to secrete a substance, secrete a secretion, has various chemicals and enzymes that break down the organic matter around them, and then they can take this up into their body, into the mushroom itself. So what if we use these against cancer? In previous results in the lab, we've been able to detect a direct cytotoxic effect against cancer cells by using mushroom secretions. So in my specific experiment, utilized cell culture that in a sterile hood, I would remove my acute lymphoblastic leukemia cells from an RS411 cell line from the incubator. I'd bring them into the sterile hood, and then I would remove the media that they're in because they need a growth media to grow. And then I would re-inoculate them in new growth media so they can continue to grow. And one thing that was very interesting about this specific cell line is previously in the lab, we've dealt with adherent cells. So this is a cancer cell that's on the bottom of the 10 centimeter dish. Meanwhile, these cells, because they're a blood cell, they, thank you so much, they actually float within the media. So what did I do with my mushrooms? So my mushroom mycelia, I grew them on wood, potato, dextrose, yeast, agar, and as they grew, once they reached the end of the plate, we were able to remove these secretions, filter them, freeze dry them, then bring them to a 20x concentration, as you can see right here. So in the setup of my experiment, I used a 96 well plate and a cytotoxicity assay. As you can see here, I had 6 different quadrants, and each one of these quadrants, each well had 20,000 cancer cells. And it was given against the control of water versus an amount of the drug. And we started here with 2 microliters and went all the way down to 12 microliters. And this was done in replicates of 5 for validity of the study. So let's discuss the results. Results were actually very interesting. As you can see here, as there was an increase of 2 microliters of drug, there was a distinct increase actually in cell viability, up to 700%. And this actually indicates similar to results what we've seen with healthy cells and not the cancer cells. So this begs the question, is it because we're dealing with a cell suspension where the cells are floating within the media, or is it the secretions themselves? So we went ahead and did an NGC chromatography system, and we're able to isolate what appears to be 3 different glycosylated proteins. One that was 6 kilodaltons large, one 0.65, and one 0.5. So let's discuss these results. Well, first, we're dealing with the secretion profile. Why may have it increased the cell viability instead of decreased? Well, in previous uses of Lentinus adotes, the secretions have had 1 glycosylated protein. Instead, in our experiment, we ended up having 3. So the question is, when looking at these 3 proteins, could potentially 1, 2, or 2 of them actually have cytotoxic effects? That's where further experimentation would need to be done. Potentially, this one itself could have been leading to the cell proliferation. We also are dealing with a suspension cell line. So a new cell line we have not previously dealt with before in this experiment. So potentially a greater concentration of the drug would be required in order to kill the cells. As well as one hypothesis we have is that in the adherent cancer cell to the bottom of the dish, that these type of cancer cells have a receptor that the secretions release a protein that bind that receptor that cause cell death. But these floating immune cells do not have this receptor. So what are some future directions for research? Well, we can isolate these 3 fractions through the NGC chromatography machine, and we can use these glycosylated proteins against the ALL cells and see which of these has a cytotoxic effect. We also can take different medicinal mushroom secretions against ALL and see which ones are effective and which ones are not, as well as we can take these same secretions and use them against different cancer types and find the results. As well as we'd like to be able to replicate the conditions of my experiment and see if we can get Lentinus adotes to release the same secretions again. All right, I want to give a huge thank you, first and foremost to God for being faithful and loving me and bringing me to this point. I would not be here if it wasn't for his love and his faithfulness. I want to give a huge thank you for my family for supporting me all of these years, and I wouldn't be here too without their help. And Dr. Ranahan for his leadership and for his advice and answering my many questions, and Diego, who presented before me, the lab manager, for also answering my many, many questions, and also for all of you, my classmates and my friends, for supporting me all this time. I'm just immensely grateful. So thank you so much. And I want to ask, is any questions at all? Yes, of course.
So I see with 280 you were looking for levels of protein. What were you looking for with 340 and
So with 340, this is why we believe it's potentially a glycosylated protein. So glycosylated protein, we're dealing with a protein with polysaccharides, right? Well, so we see the 280 nanometer wavelength, this indicates our protein. And as you can see, the corresponding peaks that are corresponding with the green, we see in orange. Well, the 340 nanometers indicates a polysaccharide. And so as we can see this direct correlation between the two, it's indicating glycosylated proteins. Thank you. Any other questions?
So you're saying in your experiment that the shiitake mushroom secretions actually increased the growth of cancer cells, correct?
Yes.
So was this a novel experiment on shiitake mushrooms, or have shiitake mushrooms secretions in the past been shown to kill cancer cells?
That's an excellent question. In previous years and experiments, the secretions from Lencinus adotes have shown a cytotoxic effect against cancer. I believe last year there was a presentation given by a friend of mine, Noelia, that involved breast cancer cells involving Lencinus adotes that showed that correlation of a decrease of cell viability. And so that's where the questions come into play. This is our first time with this cell line. Why is it increasing cell viability and up to 700%, similar to a healthy cell in comparison to a cancer cell? So this was very relevant research for the lab. So thank you. Any other questions?
Thank you very much.
All right. Thank you.